Okay, welcome to the lab test review for the reproductive system. We'll be going over both the male and female reproductive system today. Remember, do not use this presentation as a substitute for either coming to lab or going to biosis in order to review the material. This is simply a subset of the information that you're going to need to know. So we'll start out with models of the reproductive system. We'll be looking at male and female reproductive models as well as slides of tissue from male and female reproductive organs. So we'll start with the male pelvic model. We want to identify the indicated structures on the following slides. So what is A pointing to? A is pointing to the vas deferens. The vas deferens. This is the cord that allows sperm to move from the epididymis, shown here, all the way up through the spermatic cord, inside which is contained the vas deferens, through the vas deferens, and eventually to join the urethra at the ejaculatory duct and then be expelled to the outside environment. Okay, B is pointing to the spermatic cord. The spermatic cord is an assembly that contains both the vas deferens and the testicular artery, veins, nerves, and lymphatics. Okay, so what's the name for this entire assembly? It drops down through a special hole in the lower abdominal pelvic cavity called the inguinal canal, guided by a tendon in the scrotum down here called the gubernaculum. As the descent of the testes completes, what we've achieved is the location of the male gonads, the testes, outside the body cavity, and this temperature, which is 2 to 3 degrees below body temperature, is optimal for sperm production. Sometimes males are born with undescended testes, and this is a condition known as cryptorchidism. In this case, the testes have to be rescued surgically, brought down into their scrotal location, and then sperm production generally proceeds normally after that point. Okay, C is pointing to the seminal vesicle. D is pointing to the prostate gland. The seminal vesicle and the prostate gland both produce components of semen. E is pointing to the scrotum, the sac inside which the testes rest. F is pointing to the shaft of the penis, and G is pointing to the glands of the penis. Okay, this is a zoom up here, and what you're looking at is the male reproductive system. Specifically, we're zooming in on the area just under the urinary bladder. A is indicating the prostate gland. B is the prostatic urethra. Notice that the prostatic urethra runs through the middle of the prostate gland, and the danger, of course, with this is that occasionally the prostate gland can become inflamed and can occlude this region of the urethra, and the result then is that urine finds it difficult to pass. Generally, this is fixed by coring out this region of the urethra to allow the free flow of urine. This condition can occur in conditions such as prostate cancer and prostatitis. Okay, C is pointing to the seminal vesicle, and C is pointing to the ejaculatory duct, and D is pointing to the seminal vesicle, okay, which also contains components of the semen. The ejaculatory duct represents the point where the vas deferens joins the urethra, okay, and this is how semen is able to exit the body, okay. So again, C, the ejaculatory duct, and D, the seminal vesicle. Okay, moving on. This is a pullback, okay. A is pointing to the corpora cavernosa. C is pointing to the corpora spongiosum. Now, what are the important features of these pieces of tissue? They're important in the erectile response. What happens when an erection is achieved is that these tissues engorge with blood and the penis begins to stiffen. B is pointing to the epididymis. This is where sperm learn to swim. D is pointing to the tunica albuginea. This is the white jacket that encloses the testes. Okay, the testes are underneath this region. Tunica albuginea is Latin for white jacket. white jacket. E is pointing to the penile urethra, and F is pointing to the glands of the penis. Okay, now moving on to the female pelvic model. A is pointing to the round ligament. This is one of the stabilizing ligaments of the uterus. B is pointing to the clitoris. C, the labia majora. D, the labia minora. E, the urethra. Or I'm sorry, E, the uterus. And F pointing to the fallopian tube. Okay, so the significance, notice that the uterus leans forward over the urinary bladder. This is the urinary bladder. As a result, pregnant women often feel a greater urge to urinate due to the increased pressure on this particular organ. Moving on, we're zooming in here on a sagittal section through the female pelvic region, and we've removed some of the overlying structures from the previous slide. A is pointing to the ovary. B is pointing to the clitoris. C is pointing to the labia minora. D is pointing to the labia, C is pointing to the labia majora. D is pointing to the labia minora. E is pointing to the vagina. F is pointing to the cervix. And G pointing to the uterus. And finally, H is pointing to the fimbriae. Okay? All right, now we're going to zoom in on the uterus. A is pointing to the myometrium. This is the smooth muscle layer that's in the middle of the organ that aids in the expulsion of the baby at birth. B is pointing to the endometrium. The endometrium is that layer of tissue from which the baby derives nourishment via the placenta. It's generally engorged with blood, and this is how the baby gets its oxygen and its nutrients, because obviously it cannot feed. C is pointing to the cavity of the cervix, or the cervical canal. Cervical canal has two openings, an external os located here and an internal os located here. Okay, and in between, the cervical canal exists. Okay, this is a further zoom up. A, again pointing to the round ligament. B, the fallopian tube. C, the ovary. D, the fimbriae. And E, again pointing to the uterus. And again, notice the urinary bladder down here. Incidentally, this is the pubis. This joint where the two pubi meet medially is known as the pubic symphysis. This is the joint that has to relax in order for the baby to crown at birth. Okay, next we're going to be looking at some reproductive slides. Okay. The question is, from what organ are each of these slides taken? Well, 1 is taken from the seminiferous tubules. 2 is taken from the vagina. 3 is taken from the ovary. And 4 is a cross-section through the penis. This is the urethral opening. These are the corpora cavernosa. This is the corpora spongiosum. Okay? And for those of you wondering what 3 is indicating, this is an ovum, and this whole structure inside which the ovum sits is called a follicle. This follicle is a mature follicle, or Graafian follicle, so it is nearly ready to ovulate, to allow the egg to spring forth and be sucked into the fallopian tube. Okay, for each of the next slides, identify the organ from which the tissue came. Okay, A is indicating a seminiferous tubule coming from the testes of the male. B is pointing to the lumen of the seminiferous tubule. C is pointing to the interstitial cells between the seminiferous tubules. Interstitial cells indicated here. And what you're looking at basically is the birthplace of sperm. Okay, the cells at the periphery are called spermatogonia, and what they do is divide mitotically, giving rise to primary spermatocytes, which enter meiosis, producing secondary spermatocytes at the end of meiosis 1, which then complete meiosis and form spermatids. The spermatids eventually will mature into spermatozoa, and they will be propelled by peristaltic contractions all the way to the epididymis, where they will learn to swim. Okay, next question. What are the cells indicated by the arrow? and those are spermatids. Spermatids. Next question, what are these cells pictured here? These are sperm. The arrow here is indicating the head. This is the tail. And this area here would be called the midpiece. This is where the engine exists that allow the sperm to propel themselves. Okay. The head also contains a very important structure known as the acrosome. The acrosome. The acrosome contains powerful digestive enzymes that allow it, once it meets the egg, to burrow its way through the outer layer of cells that cover the ovulated egg, known as the corona radiata. Eventually, these sperm will drill down to the zona pellucida, and once they pass through that region, they will fuse with the egg cell membrane in the act of fertilization. Okay, this is a cross-section through the penis. Okay, A is indicating the corpora cavernosa. This is on the dorsal aspect of the penis. Notice that you've got two of these. B is pointing to the penile urethra, and C is pointing to the corpora spongiosum. Again, the corpora cavernosa and the corpora spongiosum are erectile tissue. They engorge with blood when the male has an erection, causing the penis to stiffen. Okay. Next, we have a peek at some ovarian follicles. Okay. And what we want to do is identify which follicle is which based on the terms that we've been given here. And so if we go to the next slide, you can see the appropriate labels for each of these types of follicles. And what you're looking at here basically is a developmental sequence. Okay. Again, we start with oogonia early in birth that divide mitotically. These lead now to primary oocytes, then to secondary oocytes, and then finally to a mature ovum. Notice that in the female that for every oogonia that undergoes meiosis, we only generate one ovum, and the reason for that is that at meiosis 1 and meiosis 2, we divide unevenly, producing a cell called a polar body, which is shed and discarded during the course of oogenesis. Okay, so what you're looking at here is the progression of the follicles, and this also applies to the names of the eggs that sit inside them. The significance of this progression is that once the follicle is mature and has this fluid-filled antrum, it becomes very fragile, and the action of the fimbriae sweeping over the ovary will cause the Graafian follicle to rupture and release the ovum with its corona radiata, these follicular cells cloaking it, and it will then enter the fallopian tube. The significance of the corona radiata is that it protects the egg from the female's own immune system so that she doesn't attack and destroy her own gametes. Okay, next question. Identify the follicle types indicated by the arrows and indicate the tissue from which this, the organ from which this tissue was taken. Again, this was taken from an ovary. Okay, A is indicating primordial follicles near the cortex of the ovary, and B is indicating now a Graafian follicle, and we can tell that because of the tremendous amount of fluid inside the follicle. The ovum is here in the center, and this follicle is nearly ready to rupture. This is just another slide, again, from the ovary, and we can see here A indicating a primordial follicle, B a secondary follicle, and C indicating a mature or a Graafian follicle. Okay, next question. Identify the follicle type indicated by the black arrow. This is a Graafian follicle. We know this because of the large fluid-filled antrum. This is the ovum here, indicated by the blue arrow, and this again is the corona radiata. Okay. Next question. Identify the follicle type. The black arrow again is indicating a Graafian or mature follicle. The cell indicated by the blue arrow is the ovum. That is the egg. Okay. All right, next question. From what organ was this tissue taken? This was taken from the vagina. A is indicating the mucosal layer, and B is indicating the submucosal layer. So this is over here a high magnification, all right, and this is low magnification. Okay, and so you can see the stratification here. Notice that these tissues are stratified squamous here at the top, and B is indicating the submucosal tissue, which generates the secretions that protect and lubricate the vaginal canal. Okay, moving on. Identify the layers indicated by the arrows. Okay, the blue arrow is indicating endometrium. This is taken from the uterus. And red is indicating the myometrium, and this is where the smooth muscle tissue exists that is eventually going to allow the uterus to expel the baby from the birth canal. Okay, that brings us to the end of this particular lab test review. Again, remember, please attend lab or go to Biosis in order to comprehensively review all this material. Do not use it as a substitute for actually interacting with the models and with the slides. And I will see everybody in class.